Well, hi, everybody. Thanks for joining us tonight. My name is Patrick Murphy, and I'm the Executive Director of the Saranac Lake Area Chamber of Commerce. And I'm really excited for us to be able to have this panelist of well-respected and experienced experts in vaccines, in immunology, in microbiology to participate in this community conversation about getting vaccinated for COVID-19. Really happy to be able to have them here with us today. I just wanted to take a moment to introduce myself. Again, my name is Patrick Murphy, and I'm the Executive Director of the Saranac Lake Area Chamber of Commerce. And the Saranac Lake Chamber likes to help convene the community around important conversations. We feel it's important to build fellowship through social functions and community events. And we try to convene conversations that are important to our members and to the community. So this is just one effort in which we are trying to pull important conversations together that are important for you. And again, we're really excited to be able to have these panelists here to help answer your questions. Before we move on, I just want to thank our co-hosts for tonight, North Country Community College, the Malone Chamber of Commerce, and I'd like to introduce quickly Matt Courtwright from the Ticonderoga Area Chamber of Commerce to say a couple words.
Thank you, Patrick. And Patrick said a thank you to our panelists. We're excited to be partnering North Country Community College this evening, and we're very appreciative of Patrick and the Saranac Lake Area Chamber of Commerce for their continued support and collaboration throughout the COVID-19 pandemic. So thank you for everything you're doing in the Saranac Lake region. We appreciate you.
Thanks, Matt. Yeah, it's been great how our two smaller area chambers have been able to team up and pull off some great events. So I appreciate your collaboration over the past year. Has been great. Before we get to our panelists, I just want to offer a little bit of guidelines for how we are able to answer questions for tonight. Please provide your questions at any time during the session. You can do it via chat, and you can do it on, if you're listening in through Facebook, you can also do it in the chat on Facebook, and we'll try to be able to track that as we go along. So feel free at any time to provide a question. If you have something on your mind with what the panelists are talking about, please provide that in the chat, and we'll get to it as they come in. This is definitely a conversation. So we want this to be a two-way street. We're gonna leave the mics and the videos off for participants just for ease of for viewing and for being able to hearing each other. But please utilize that chat function so that we can get to your questions. And we're here with no specific agenda to really push anything one way or the other. We just want to be able to get questions to you that you might have regarding getting vaccinated. So with no further ado, I'd like to have Sarah Shoemaker, one of our panelists, be able to introduce herself.
Hi, everybody. I'm Sarah Shoemaker, and I guess my sort of qualifications to be here are I have a bachelor's in biology from Washington University and a master's degree in cell and molecular biology from the University of Pennsylvania. I've spent time in the past doing research at the Trudeau Institute, at the National Cancer Institute, and for the past 10 years, I've been teaching microbiology, biology, and chemistry at North Country Community College. So I'm excited to be here. Thanks for having me.
Hi, everyone. My name is Dr. Eric Yeager. I'm an associate professor at the Albany College of Pharmacy and Health Sciences. So similar to Sarah, my background is in immunology and infectious disease. I have my PhD in immunology from SUNY Albany. I spent time at the Trudeau Institute from 2004 to 2008 researching influenza viruses and the effects of aging on the immune system. And I see some of my colleagues in the audience, so great to see you again. And in the past, I've been teaching courses on viruses, microbiology, and immunology. And over the past year of the pandemic, I've done a lot of outreach trying to educate the public about the pandemic and about the vaccines, and I'm happy to be here tonight.
Hi, I'm Chris Campersher. I am an immunologist. I got my PhD from Loyola in Chicago, and at that time I studied immune responses against viral infections. And then I went on from there to a postdoc here in Saranac Lake, and loved it here. And it was here about 5 years. Similar thing, I was studying immune responses, largely in the context of viral infections. I then went on to a position at Pfizer. I was there for about 13 years, doing drug development of mainly cancer therapeutics and also vaccines, though. And towards the end of my time there, I was involved in the efforts to develop the COVID-19 vaccine at Pfizer. So it was only just this past September that I moved back up here. I took a position at Bionique as their chief scientific officer. And so, yeah, I moved back here because I love the area, I love the town, and I love the people. So I'm really thrilled to be back here and glad to be part of the panel.
Well, that's one thing that I forgot to mention about before I introed the panelists, and that each one has a connection to Saranac Lake. And I think that is kind of just a neat factoid to talk about how Saranac Lake has these type of connections with these types of experts. And it makes me kind of proud to be here in the Saranac Lake area and to do what I do in my role here at the Chamber, to be able to interact with folks at this type of level. So that's really, really neat. And it's just kind of a cool factoid that I think I wanted to reiterate, how each of these folks have a pretty strong tie to the area. So let's just jump right into the first question, which we fielded a bunch of questions ahead of time. And I wanted to just start off with some of those questions that came in from folks ahead. And there's a first question just about what are some of the potential risks for getting one of the vaccines. And Sarah, we'll just start with you since we started with you introing. So Sarah, if you want to just provide, some feedback and perspective on what are some of the potential risks for getting vaccinated.
Sure. So the risks that are common to most vaccines are, of course, soreness and pain at the injection site. Another very common response to vaccines, but particularly to the Pfizer and Moderna vaccines, is these sort of prodromal effects that you get, kind of the way that you feel right before you get like a bad cold and you get the headache and a fever and just kind of achy. Those are all signs of an active immune response. The way that your body reacts when your immune system is ramping up and you're secreting all of these cytokines, these inflammatory molecules, and you're really producing a strong immune response. And that's very true with these vaccines. Very common, especially after the second booster. Some of the more serious side effects that have been seen, there have been a handful of cases of anaphylaxis following vaccination. Anaphylaxis is a severe allergic reaction, and this happens all the time to foods, to other drugs that people take, medications, antibiotics. The good news is that with the vaccine administration, although it's a very rare side effect, there have been a handful of cases of this over the course of millions of vaccines. But it's something that practitioners are aware of. So they watch out for it. They monitor people. You have to stay for 15, 30 minutes after you get vaccinated to keep an eye out for that response. It's very immediate. So if it doesn't happen immediately, it's not going to happen, and it's very treatable. Other side effects, the Moderna vaccine can cause a red rash a few days after the shot around the site. That's kind of, it just actually looks kind of like a bullseye. It kind of looks a little bit like, for those of us up here who have seen Lyme before, but it's not, it's just, they call it Moderna arm, I think. And that goes away as well. That's really it in terms of a lot of people ask me about long-term effects. I might be jumping the gun and carrying out this answer, but there's no precedent for a vaccine to cause long-term effects. And even though we don't have the longitudinal data to say whether or not this vaccine could cause long-term effects. There's no other vaccine that ever has. And so there's no reason to believe, and there's no biological mechanism by which these vaccines would cause long-term effects. The mRNA ones especially are very unstable. mRNA is a very unstable molecule and degrades very quickly in the body. So the idea of it causing any long-term effects is not very biologically plausible. So most of the risks that you're seeing are risks of feeling crappy for a day, missing a day of work, maybe a couple of days if you have particularly rough side effects, but that's it.
I think I can add on to what Sarah mentioned. Is much of what Sarah mentioned was kind of the side effects. In terms of risk, I think it's important for people to appreciate too that yes, there's been some news stories, particularly with the AstraZeneca vaccine, and at points with the Pfizer and Moderna vaccine, is that these vaccines have been tested pretty rigorously. I'm sure Chris can give us more information about that. But they've gone through safety testing, and the incident rates of severe adverse effects that Sarah mentioned are very low. Basically on par with vaccines that have currently been approved. Again, these vaccines have been authorized, or not fully FDA approved, but the rates of severe reactivity is not anything different than the vaccines that are out there. I think what we see is because they're under increased scrutiny, when there is an effect, like potentially a link between blood clots or a severe allergy, it brings pause, because it really is, in all the media sources, you're like, wait, is this the vaccine is new? Is there something different about that makes it more of a risk? And I think the other thing to keep in mind too is that vaccines are given to healthy people. So they are clearly tested to make sure we don't want to cause harm to a healthy person by and large. And because of that, the companies look carefully at risk versus benefit. So overall, we know that there are extreme risks with COVID-19. It is a severe disease. You can be hospitalized. It can be fatal for some individuals. And we're just beginning to understand the long-term complications due to that infection. So if you compare that to the vaccine, the vaccine is extremely low risk. Now, again, there are certain populations for a variety of reasons cannot get vaccinated. So the CDC and some other websites have a great place to look for information. If you're someone that maybe is, for example, if you have a certain health condition, or you're immunocompromised, like a transplant individual, or on cancer therapy, and you're uncertain about the vaccine, definitely your doctor is a first place to start. But there are certain individuals where a vaccine, irrespective of what's a COVID-19 vaccine or a traditional vaccine, may not be best for that person. So in that case, we have to depend on others getting vaccinated. And we'll talk more about herd immunity, I'm sure, in another question.
Well, perhaps this might be a good moment for Chris to jump in and talk about those safety, how the vaccine was tested for safety. Sarah did a great job explaining about the potential side effects, and Eric expanded a little bit about how that vaccines are tested for safety. But Chris, could you provide some insight on how, with the way in which they sped up the type of production to get the vaccine out, how they tested for safety?
Yeah, so I think it'll help if I just share a slide really quickly. So I think this will help illustrate that concept there. So I put this slide together that shows basically the traditional approval pathway for vaccines here on the right-hand side, and then the emergency use authorization pathway that has been used for COVID vaccines. And just very quickly, I mean, after, once you've developed and created a vaccine and gone through preclinical testing, then as long as, in this case, it's the FDA, because we're talking about US, but then once the FDA approves to say that, yeah, you can go ahead into clinical trials, then the clinical trials proceed in 3 phases, phase 1, 2, and 3. And the main thing is that, so phase 1 is small numbers of people, and it's really intended just for safety, to understand whether the vaccine is safe. And then you ramp up the numbers of people. And then phase 3 studies are usually thousands of people or more than that. And it's very often much more than that. And then, that's the data that you use then to submit to the FDA. The FDA will review that. And then, after a certain period of time, they will vote and then decide to whether to approve it or not. And that usually takes about 10 to 12 years. That's a typical time frame for being able to develop a vaccine. So the COVID vaccines were developed essentially in about a year, which is incredible if you think about it. So, I think some people have concerns that, well, if it's that fast, are we kind of skimping on the safety evaluation? And I think the answer to that is no. And I can tell you the main reasons why this got accelerated. So first of all, just to make the point, it is exactly the same process. So nothing has changed about the process for testing or for evaluating the vaccine. It's all just been incredibly accelerated. So the first reason for that is once we created the vaccine, so like, for example, at Pfizer, we were testing 3 different versions of the vaccine all in parallel. We normally wouldn't be doing that just because you'd want to spend more time in the early stages to get a better sense for which one was going to work before you spent all that money on doing clinical trials. But because of the situation, we decided to go ahead and test 3 separate vaccines in parallel. The second thing is that the testing, the clinical trials were going on at the same time we were scaling up manufacturing. Okay, so this is large scale manufacturing to provide enough doses. And normally, again, that wouldn't happen. Normally you would get well into the clinical trials and really towards the end of the clinical trials before you were confident enough to say, yeah, okay, we're gonna go ahead and put all the resources into the large scale manufacturing. And the third thing, and I think this is probably the biggest factor actually that sped up the clinical trials so much, is that you had massive numbers of people that were getting infected, right? So very often when you're testing a vaccine, you have to wait. You have to wait until you have enough people infected so you can get all the data that you need to be able to understand whether it's actually working. Whereas in this case, we had huge numbers of people very, very quickly that became infected so that we could very quickly get enough data to understand not just the safety, but also the efficacy. So that's really what sped it up. But otherwise, then you still have the same FDA review. And then in this case, it's an emergency use authorization that allows it to be released to the public for for use. But it's essentially the same process, and the formal approval for it will come later on. So I think that's the main point, though, is that the safety evaluation of the vaccine basically was done in all the same ways. It was just greatly accelerated. So I'll just stop sharing.
Well, that's pretty interesting. Is coming from an experience where you were actually working on one type of vaccine, Chris. Sorry to go right back to you, but should folks be worried about using one vaccine over the other from a manufacturer, from whoever manufactured it?
So my answer to that would be no. A general no. The Moderna and the Pfizer BioNTech vaccines, those are both very, very similar vaccines. So they're both RNA vaccines, and they're RNAs that have the genetic code for just one protein from the virus, the spike protein that's on the surface of the virus. The J&J, Johnson & Johnson vaccine is a little bit different. It's just a different way to introduce that genetic code for that same spike protein into cells. So instead of so the Pfizer vaccine and the Moderna vaccine are basically RNAs that are encapsulated in this kind of lipid, sort of fatty cage that protects it from being degraded, but also allows it to get into cells so that it can, trick the immune system into thinking that it has an infection in the cells. The Johnson & Johnson vaccine, it's the same approach overall to get this genetic material in the cells, but it's a adenoviral vector. It's not a lipid nanoparticle. It's an adenoviral vector, and then the genetic material comes in the form of DNA in that scenario. But essentially, they're all getting at the same goal, which is to force cells to express this spike protein so that it looks like you've got infected cells, and then that fools the immune system into responding against it, and then generating a good immune response, so that when you get eventually challenged with the real infection, you've got a response there that can eliminate it before it causes any symptoms.
Sarah, do you have anything to add about knowing of the different available vaccines and getting one that's offered to you over the others?
I have a slide for this. Hold on, see if I can share mine.
Could it be outdone by Chris? Is that how it was? You had a, yeah.
Okay. How do I make this full screen? Is this working? Grant access. Sorry, okay. I don't know if this is too much. So one question that sometimes people have is, what are the ingredients of the vaccines? So this is a document from the CDC. This is all the ingredients in the 3 different vaccines available. So here's the Pfizer, the Moderna, and the Johnson & Johnson or Janssen vaccine. So Pfizer and Moderna are there. I tell people it's like taking Motrin versus Advil versus generic ibuprofen. They're essentially the same active ingredients. They're both mRNA vaccines. They both code for the spike protein on the surface of the virus. And then all the other inactive ingredients are just sort of fillers. And mostly I've pointed out what all these big, scary named words are emulsifiers, which are commonly used in food and cosmetics that help to disperse or dissolve the whole product and form these liposomes. So when Chris was talking about these sort of fatty spheres or bubbles, they look like this. All right, these are the nanoparticles that we're talking about. Nano just means, oh, I should know this, 1 billionth, 1 trillionth, 10 to the ninth, 10 to the negative ninth. So it means very, very small, okay? And that's what we mean by nanoparticles. They're very, very small particles made up of these lipids, and they can encapsulate materials. So they're the delivery system. They're a vehicle to deliver the mRNA to the cells. Cells are made up of phospholipids, of fats, and so these things can fuse with cell membranes and help deliver it to cells. The other ingredients on here, most of these are just salts. They're electrolytes, which are important because when you inject something into the body, it needs to match the concentration of the body fluids. So it needs to have the same salt and sugar concentration. That's what the sucrose is down here. And then some of these are ingredients that serve as buffers, so they keep the pH steady. There's no heavy metals. A lot of times people are concerned about mercury and other heavy metals as ingredients in vaccines. There's no fetal cells. There's no preservatives really at all, except if you count the salts as preservatives. It's one of the reasons why they have to be stored so cold and have such a short shelf life, because they are actually fairly preservative free. And mRNA in particular is a very unstable molecule. It degrades very easily and very quickly. So the difference, I suppose, that in that active ingredient, I think Chris covered pretty well. It's just slightly different. It's not RNA, it's DNA, and it's covered in a viral protein shell. And that's just the delivery system that vaccine uses. So they all are excellent vaccines. There's a lot of, I'm probably jumping the gun here a little bit, going to bring up efficacy a little bit between these. So you may have heard that the Pfizer and Moderna vaccines have somewhere in the 90, 95-ish percentile efficacy, meaning that most people, 95% of people, you have a 95% chance of not getting sick if you get that vaccine, versus the Johnson & Johnson efficacy trials were slightly lower. I think they were in the 60s, 68, 64, somewhere in there. But something to consider, something very important to consider about that is that the Johnson & Johnson trials were done later. They were done later in the outbreak when we already were seeing these new variants come up, Brazilian, South African, the UK variants, which are all more contagious. They were also done in other countries. So Moderna and Pfizer were both done in the US, and Johnson & Johnson was done in Brazil, in South Africa, in countries where these variants arose. And so it was basically a trial that was in a way more stringent, a stringent test against these variants. And the important takeaway is that it still had 100% protection from death from coronavirus. All of these are equally protective against hospitalization and death. So while all of them may still leave you susceptible to getting coronavirus, your symptoms will be much, much less. And all of them so far have shown to be incredibly protective against the most serious outcomes, which is the ultimate, most important goal.
Well, Sarah, I appreciate that, and I appreciate this slide. It looks like someone in the room asked if they could get this information directly from you. So I think the easiest way is I'll leave my email in the chat, and you can share your info to me, and we could be able to dispense it. If someone emails me, I can share the information that you folks have put together to share with the room. And yes, this recording will be available for folks to be able to share with their friends. So we can make sure that that's available as well. Eric, I'll come to you now to talk about kind of where Sarah left it off, which was, choosing if you knew a certain state site was giving out one vaccine or the other, should you feel, more inclined to go and get it? But also I think she was talking about some other aspects of the vaccine, which is, those efficacy rates, and you hear those numbers and you wonder, exactly how they change between the vaccines. So what might you add to this part of the conversation?
Yeah, happy to jump in. And Sarah did a great job kind of bridging into that idea of efficacy. And I know we see those numbers and we kind of think of grades on a paper, like you definitely want an A+, you don't want that C, right? So you see those numbers and it might give you pause about why can't I get a certain vaccine? And currently, I believe in New York State, you don't really have a choice. You go to the vaccine site and what they have available, they'll give. And probably right now, it's predominantly the Pfizer and Moderna vaccine since the J&J vaccine just rolled out. I think what's important to realize about what vaccine efficacy is that's a number that was obtained from the clinical trial. So it's done in a very controlled setting, as Sarah and Chris had talked about. So it does give us guidance about how protective vaccine is. But of course, when you bring it out to the larger population, there's a lot of variability in terms of individuals' age, their gender, their genetics, if they're on medications, their health status. So it can differ a little bit. And I think the most important thing to think about with this efficacy number, as Sarah hinted, is that all three of them, and there are 2 additional vaccines probably forthcoming in the next month or 2, all these vaccines look to prevent very well against severe disease and prevent against fatal disease. And I think that's where we have to be looking for is if you're given a vaccine, take it. The vaccine does better for you in your arm rather than on the shelf. And they're all protective. And what we really do is, our goal is to get this pandemic to the point where it's not any different than kind of like the flu season or the common cold. So yes, we have to worry about that. You can get sick from that. But if we have a vaccine that can prevent those that are vulnerable in our populations, throughout the North Country, there's a number of individuals that are elderly or have pre-existing health conditions. We can get the vaccine levels to where we need them to be, then those people are protected. Again, protected to the point meaning they don't have severe disease, they're not hospitalized, and there's no worry about complications and fatal disease. Oh, I think you're muted, Patrick.
Darn it. I was hoping I could get one session without being muted while I talk. Since you're talking about how, if it does better to get the vaccine in your arm than having it on the shelf, and getting the right number of populace to what was alluded to earlier in the session, which is herd immunity. Could you go into that and perhaps provide a little bit of just insight into what that means? And how quickly do you think we'll be able to get there?
Sure, happy to. I think the one exciting thing about the science, that Chris and Sarah talked about is that, think where we were a year ago. We were all living in fear about this pandemic, uncertain what it was going to mean. And here we are now. We have 3 vaccines available with, maybe 2 more on the horizon. And to have all these effective vaccines that are able to be given to people, accessibility is increasing, availability is increasing. So the more people we get vaccinated, there's 2 benefits. The more people that are vaccinated is going to provide herd immunity. What herd immunity simply is the more people you have protected against a disease, either by infection or vaccination, prevents that disease from spreading. So again, the virus is trying to get from person to person. And if all those people in the room are vaccinated, the virus isn't going to travel around very far. And same as in a community. So we want to get to that level where the virus can't travel as easily from person to person in the community. And what that level is kind of still somewhat of an active area of modeling and mathematics. It's thought to be in the area of maybe 80% or so. So we think about how many people have been vaccinated thus far. And I just looked at the data, I just pulled it up recently. Right now, we've administered 127 million vaccines in the US. So you're like, well, it's an incredible number. That only gets us right now to individuals that are fully vaccinated. We're at 13% of the population. And that doesn't, of course, count people that were previously exposed and have some immunity, but we're at 13%. So think about that 13%, we have to get that 80%. We still have a lot of work to go. So we need to get more people vaccinated to get them comfortable with the idea of being vaccinated. And that's why an event like this is great to kind of educate and inform, and particularly to people out in the audience, when you go back to your families and friends to kind of have these conversations. And one of those important facts about why should I get vaccinated? And the sometimes I hear is, well, I'm healthy, I'm young, I don't need to be vaccinated. I can kick COVID's butt, I don't have to worry about it. And, one, we don't know COVID can infect individuals of all ages, from children to very old. And secondly, we know too that natural infection may not be as protective as a vaccine. And getting to that herd immunity that you mentioned, Patrick, is I think the best route to get there is through vaccination. And currently with our 3 vaccines we have available, if you're able to get an appointment at those clinics, if it's 2 shots or 1 shot, get your vaccine, you're protecting your community and you're protecting the country.
Chris, would you add anything to that? Or also, the next question kind of leads into, from what Eric was talking about, to folks who already have been infected by the virus, or who were asymptomatic, tested and were asymptomatic. Why should they get the vaccine? Do I need to get the vaccine if I've already had it? Could you talk about how folks in the population add to also herd immunity, but should they be also vaccinated?
Yeah, so what I would say about that is that if it were totally black and white, where you had 100% protection in everybody that was vaccinated, then or that was recovering from an active infection, then yeah, you might be able to say, well, you really don't need to be vaccinated because you're probably protected, but that's really not the case. So a couple things on that. One is we know in some people, we do see, even after natural infection, we see the immune response starting to wane. And so, you may not be completely protected against subsequent infections. And then maybe just as importantly, you can become infected and maybe you're not going to have any symptoms at all, but you could be transmitting that virus to other people, right? And the reason for the fact that you have waning immunity over time, at least in some individuals, isn't really that clearly understood. One interesting thing is that there are known proteins, one in particular, that within the coronavirus, that is known to impair the ability of the very early immune response to happen. And it's possible that impacts longer term immunity after you've had your immune response to that virus and hopefully cleared it. But that's that's just one possibility. So that's natural infection. But if you get vaccinated, you're just putting in this one protein that you're intending to make an immune response, and that's that protein that sits on the outside of the virus, this spike protein. And in that scenario, you don't have, if it's vaccination, you don't have any of these other viral proteins that could potentially interfere with immune responses. And so the expectation and hope is that the immunity that you generate through vaccination might actually be better than what you get with natural infection.
Thanks, Chris, for explaining that. That's always been something on my mind about how, even though you might have had gotten it, your immune response has been shown recently that it is waning, and how you need to be able to be as proactive and be able to get that vaccine, because that helps boost, that type of response rate. There's a really good question in the chat about how.
Just quickly to wrap that up, I mean, that's why I think it's important to be vaccinated, even if you've already had infection, because the immune response is variable in people, right? Quite variable in different individuals. And you may have a good enough immune response so that you prevent any symptoms from a reinfection with yourself, but you potentially could become infected and then spread it to other people. So I think that's really the critical reason why you should be getting vaccinated.
Okay, thanks, Chris. There was a question here. There's two questions that I want to get to that's in the chat. One is, what would you say to an anti-vaxxer? But which I think is a really interesting question about how we can take some of this information from the folks who are hearing this in the room and then be able to communicate that to other people. But there's a question that's been passed around in someone's household here about whether being vaccinated prevents you from spreading COVID, or once you're vaccinated, can you not transmit the virus? And I think Chris touched on that a little bit. But Sarah, I don't know if you have a response to, elaborate a little bit about what Chris was talking about at the end there.
I do, and I also wanted to piggyback on what Chris was saying. So
great.
Your risk for transmitting the virus is way, way, way lower if you're vaccinated, because basically you become a hostile host to the virus. And so you could potentially, if you had contact with someone who was infected and you were at close contact with them, you could pick up a viral load and be transmitting that. You can, healthy people can always be carriers of pathogens, but you're much less likely to be one, and you would be one for a much shorter amount of time if your body was effectively fighting it off. So my visual that I wanted to share about, am I sharing?
Yeah, you're sharing.
Okay, hold on. I
just gotta say, Chris is being put to shame because Sarah has two visuals that she shared.
No, I like visuals. So what Chris was talking, this is exactly what Chris was talking about. So we were talking about, do people who are infected, do they need to get the vaccine? So the way that the immune response works is the first time you are infected with a pathogen, you have what's called a primary immune response. And your immune response, you make all these antibodies and you fight off the infection, but then the antibody levels drop down. And so you're not all that well protected for the second infection, but you get a second exposure and your immune system has memory. It remembers that pathogen and it starts pumping out antibody wildly. And then hopefully it prevents you from getting sick the second time. But notice that after the secondary immune response, the antibody levels don't come down all the way. They stay in the blood. And so you continue to have these protective antibodies. And that's one of the reasons why you need boosters of vaccines. One of the reasons why we do 2 shots of the Moderna and the Pfizer, because it gives you that secondary immune response and just boosts that antibody production and that memory response. And so people who have had COVID, they've had that primary immune response, but their antibody levels are waning. And they have memory, hopefully have memory cells, though as Chris and Eric had alluded to earlier, that memory wanes and the immune response seems to wane because the virus itself kind of takes the immune system out at the knee a little bit and prevents some of that from forming. So I've read that they've done some early studies and shown that people who have had coronavirus only need 1 shot. They don't need 2 because 1 shot just goes ahead and and boosts, it basically acts as a booster. The first shot acts as a booster. I've also, and I'll say I heard this in a science podcast that I listened to, which is somewhere in here. I'm gonna find it. There we go. It's called TWIV, This Week in Virology, if anybody's interested. Every week, this Dr. Daniel Griffin does a clinical update. It's about 30 minutes long. And he was referencing recently that he has several patients who had COVID and then they got the vaccine, and a lot of their long COVID symptoms are going away. So there's early evidence that the vaccines may actually have therapeutic value to people who had COVID previously. So whether or not you think you're protected from COVID, if you have any long-term COVID symptoms, there's a possibility that the vaccine can actually be therapeutic. And I just put his quote here. He always says, never miss an opportunity to vaccinate, never miss an opportunity to test, and never waste a vaccine dose. So check out that podcast if you're interested in this topic.
Eric, do you have any, final thoughts? We have a couple other questions to help transition.
Sure.
Do you have anything about, just about transmission after being vaccinated and how to wrap up this conversation before we
Sure.
Yeah.
I think just to piggyback on Sarah's point, I think there's some exciting data that vaccination could be beneficial to people that have suffered COVID previously. And I think there's some tantalizing data that maybe a single dose would work. But currently, right now, I just want to make the point that if you had COVID and you get your vaccine, you still need 2 doses if you go for Pfizer or Moderna. Don't stop after 1. Again, we still have some work to do to establish whether that's a true path to take. In terms of transmitting after you've been vaccinated, as Sarah mentioned, it makes it less likely for your body to host the virus. But the clinical studies for all these 3 vaccines just looked at disease. Remember, a vaccine can prevent infection and a vaccine can prevent disease, or do both. And right now we can just say for certainty that the vaccine prevents severe disease. To measure whether or not the virus is found in the airways, can be passed from person to person, is challenging to do. So the companies couldn't do that in those large trials. They're tracking 30,000 people. They couldn't really do that in a large study. So there's some smaller studies and some anecdotal data to suggest that vaccination does reduce the amount of virus in a person's airways that theoretically should reduce transmission. But until we have more solid data right now, that's why you see the CDC recommendations that, unless you're fully vaccinated, you've gotten your 1 dose of Johnson & Johnson or your 2 doses of Pfizer or Moderna, unless you're with another person that's vaccinated, you need to wear your mask to ensure that you're not transmitting. Because as Chris and Sarah mentioned, you could carry the virus, and we still don't know how much virus is needed to get someone sick. So even if that viral level is reduced, it might still be sufficient enough to infect grandma or grandpa. So again, we all don't like to wear masks and have to distance, but until we have a little bit more data and more people are vaccinated, it is encouraged that you can continue to follow all those interventions for safety until you have a bit more data.
Well, that's actually one of the next questions, which we were thinking, we'll get to that question about what we do when we are talking with an anti-vaxxer, because that's, I think, we talked about this ahead of time that we wanted to end the session kind of about that. So we will get to that question. But it is another question on here about, I've been vaccinated. Why do I still need to wear a mask after getting vaccinated? And, what's the reasoning behind that? And Eric, you touched on it a little bit, but I don't know if Chris, you wanted to expand on, the importance of why that's still a recommended, activity from the CDC.
Yeah, I think Eric said it very well, right? Is that we don't yet know how much virus it's going to take to infect someone else, but we do understand that not everyone that gets vaccinated is going to be 100% completely protected. So there's still some chance that you could be harboring some virus and then pass it on to other people. So that's really the reason why we need to still wear masks, at least until we get to a certain level of population vaccination.
Well, a question just came in to talk about vaccines for children and that clinical process. I'll offer it up to the group who wants to maybe talk first about what's happening currently and because I know there's been talk about for teenagers and now for younger kids too, trials starting. So is there any insight into what that process is looking like?
With these trials, and Chris and Sarah can chime in after. I'll just jump in that there's usually an age de-escalation with vaccine trials. So we start with 16 and older, 18 and older, then you work your way down. So there are trials ongoing right now for younger, young teens, maybe younger children, and eventually they'll get to even younger children. So they're starting. The data is being collected, and it has to go through that same rigorous process that Chris mentioned to make sure that, again, the vaccine is safe and as effective in children as it is in adults. But that timeline, the estimates I've seen suggest perhaps fall or end of this year when they have vaccines available for children. Which vaccine is it going to be? It's hard to say. Again, I stress the point that we have multiple vaccine platforms that we can try, and the one that is most effective is the one that they'll go with for children.
And I'll just piggyback on that and add something that Chris mentioned before, that one of the reasons the trials were so quick for these vaccines was that there were so many cases. And so with a clinical trial, there's no finite endpoint. It's just when enough people from the trial get sick that you have enough data. So it will be completely dependent on contagion and people getting sick. So in a way, you want it to be done quickly because you want to be able to get your kids vaccinated. But at the same time, you also don't want kids getting sick. So it's hard to know what to wish for, a fast trial or not. But yeah, so that's why they can't really necessarily quote finite dates, because it totally depends on people getting sick.
Yeah, I don't really have much to add to that. I know that there are trials ongoing in the pediatric populations, but I'm not aware of any data that's been released yet on those.
Okay. We'll move on to the next set of questions that we have, and they go into wondering how the different variants of COVID-19 are affecting vaccination and what might that look like for the future in terms of, there's a whole host of different things that we've talked about planning for this session, including what's that do for herd immunity, and what's that do for potential future vaccinations against, with booster shots. Sarah, would you like to start with just talking about, especially just about how variants are affecting that type of vaccination rate and what we need to know about variants?
Yes. Okay. So I'm gonna throw up a slide. It's not very pretty, but it's gonna give me my visual. Okay, so this is the structure of the spike protein of the coronavirus. And so this over here on the right is the coronavirus spike protein, and then me kind of trying to Photoshop in some various antibodies. These Y-shaped things are antibodies. So you can see that when you make an immune response, you make antibodies to all different parts of this spike protein. And when this spike protein mutates, the variants have tiny little differences at these different spots. And some of those differences make it bind to cells better and make it infect cells better. And so that makes it more contagious and more infectious. But when you get a vaccine, you're getting this whole spike protein and you're making antibodies to all parts of the spike protein. So even though maybe this part right here changes a little bit in the variant, the rest of it is still the same. And so you're still making a robust immune response. You're still very capable of making lots of antibodies that recognize this different parts of this protein. It's slightly better at getting into cells, but you still are mounting a really effective response. So while the variants are making the vaccines potentially a little bit less efficacious, because maybe some of the viruses can get into cells before the antibodies can neutralize it, the variants in the virus, it does not mutate super significantly or super quickly. And so these little incremental changes are making it a little bit more contagious, but they're not changing it so significantly that the vaccine is now not effective. It's still very effective. And so, people are still, again, with no matter the efficacy, are still not being, not dying. They're not getting so severely ill because there is still all these other antibodies that work. The other thing is that the way that the vaccines are designed using DNA and RNA means that we can make them very quickly in the lab. So it is possible that if the virus keeps mutating and changing, that perhaps, down the line, we might have a new iteration of the vaccine, sort of like every year we have a new iteration of the flu vaccine, except that flu vaccine is much more onerous to make and produce than I think these coronavirus vaccines are from a biotechnology perspective. So that gives me a lot of comfort and knowledge in knowing that if the virus does mutate so much the vaccines are not efficacious, we can simply remodel, find the recrystallize the shape of the newest dominant variant and design a vaccine to match that.
Eric, do you have anything you'd like to add about these variants and how they're affecting the,
Sure. So Sarah mentioned, so that many of the variants that were identified, and one of the things that's been important about this pandemic is the importance of gene technologies and gene sequencing. So we have the ability to detect these variants as they're arising. And as Sarah nicely showed in her diagram, a lot of these changes are occurring in that spike protein that the virus uses to get inside cells. And what's been noted is that some of these variants are in laboratory settings, but also some anecdotal data in the real world that these viruses might be better able to get inside our cells. So you might need less virus to get sick. And there's some suggestion that some of these variants could potentially be a little bit more virulent than the wild type virus that we saw early on in the pandemic. So a couple of key things about variants is, first, these variants only arise when people are infected. They can't arise by themselves in nature. They just don't spontaneously appear. So they appear when someone gets sick, the virus replicates. And like all viruses, it mutates. And as Sarah mentioned, it mutates at a lower rate than flu or HIV, but it still mutates. So these variants were not unexpected. But what is unfortunate is in some of these variants, the monoclonal antibodies that are used as therapies no longer seem to work. And in some cases, the vaccine may not be as protective in a laboratory setting. What that means in the real world, we're still trying to figure out. So kind of the race is here to get enough people vaccinated, that concept of herd immunity, because the more people that are vaccinated, there's less of a chance for more variants to appear in the future. And as Sarah nicely mentioned, the one other fortunate aspect is we have vaccine technologies now that could be readily adapted, quickly and readily adapted to these new variants. So there's possibilities, and there's been some reports that companies are trying to make these, kind of booster shots just in preparation for if we need to do that. If it turns out that this B117 or the South African variant you hear a lot about in the news, they become more predominant, or the new variant that's arising out of New York City, if that becomes more predominant, it seems like the vaccine isn't going to be as protective. And as important as Sarah mentioned, the vaccines still work, just not as well as they initially did. So there's, we still are trying to figure out what level of protection is needed to prevent severe disease, prevent fatality. So I think in kind of this era of being overcautious, the companies are just working hard, as Chris mentioned, before we get to that. You want to have the vaccine ready. You don't want to wait till you're trying to fly the plane as you're building it. We want to have that ready in case one of these variants comes out. And as Sarah mentioned with flu, I think it's probably good or bad. My assumption is that we'll be kind of thinking about COVID-19 almost like flu, that it will start to mutate like this. And we'll probably have a vaccine we'll need to get maybe every year, a couple of years. But again, if we can get it to the point where it's like the flu, and the flu vaccines we have prevent thousands of deaths and hundreds of thousands of hospitalizations a year, we're closer to going back to normal life.
Yeah, so I think I'll probably just mostly reiterate what has already been said on this, but just a couple points to make. Like Eric said, the virus is continually mutating all the time. Every host it has, it's gonna generate new variants eventually. There are many thousands of known variants that are out there, but most of those don't have any impact on the immune response. And like Eric said, if you do experiments in vitro, if you take antibodies and serum from patients that were vaccinated, you can find that some of these variants do impact the ability of those antibodies to neutralize the ability to infect. And I think probably the ones that are most concerned are, I mean, there's the B117, the UK variant that Eric mentioned. There's one out of Brazil that's also being seen in Japan, this P1 variant, the South African one. And then there are a couple actually out of California as well that have, and actually most of these share a particular mutation that seems to, make them more infectious. And then, less able to be neutralized with the common antibody responses. But, none of these viruses, though, that have been tested so far completely escape the immune response. So that's important to note. So regardless of whether the immune response is maybe slightly less effective, it still does have a big impact on the infection. And then the last point I want to make is that, these variants arise because they're continually replicating. So for the virus to do that, it needs a host. So if we can eliminate as many hosts as possible, we can really slow that mutation rate and the ability to generate these variants. And I think we're seeing a lot of variants happen or arise pretty quickly just because we have massive numbers of people around the world that are getting infected. And each one of those people, especially if they don't have pre-existing immunity, there's sufficient time for that virus to continually generate more and more variants. So, I think the best defense we have against generating more variants is to vaccinate everybody so we don't continually have all these hosts for the virus to replicate in.
Chris, could you, since we're on you right now, could you just talk about a little bit about what the future might hold? Eric touched on it a little bit about boosters and future, just 6 months from now, 1 year from now, what does that look like for either getting a booster or getting revaccinated? As if in the future, will it look like what we do with the flu now, where folks go in, certain demographics of folks go in and get a vaccine for this? Or what's it look like for us on future vaccination?
Yeah, so one clarifying thing first. So you hear a lot about boosters, and sort of historically, when we talk about boosters, we're thinking about another dose of the same vaccine. Whereas a lot of times now when people, when you hear in the news people talking about boosters, we're very often talking about generating basically a new vaccine to account for differences that are occurring in the virus. So Sarah alluded to this, where you have different variants that get generated, but the response to that can and I think will be generating new vaccines to account for those differences, so that we prime yet another area of the immune response that can handle those new variants. And that's happening right now already. Actually, all of the major companies that are producing the vaccines that are currently available are already working on follow-up vaccines to handle the variants that we know about right now. So I expect that that by the time variants are become extremely prevalent, it won't be too much after that. We probably will be able to generate a follow-on vaccine. And the only other thing I say about that is that that is one big advantage to the Pfizer and Moderna approaches, because you can generate the RNA, this messenger RNA molecule that will cover any variant that you can find in a very, very short period of time. And the way that platform is set up, you can kind of swap in and out whatever RNA to code for the right spike protein of that certain variant as much as you want. I mean, it's not totally trivial to generate another vaccine, but it is much, much faster than the traditional ways that we make vaccines. So I expect that we will be able to respond to new variants much faster with the new vaccine approaches that we have than we have been able to in the past.
Same question for Sarah or Eric.
Thanks, Todd. No, I think so. I think, like, what does the future hold? I think from a scientific standpoint, we'll have a better sense of what an immune response is to this virus. What are those correlates? Like, I'd be amiss if I didn't mention T cells, being that it was my my area of research at Trudeau. But we don't know a lot about T cells. And as Sarah had her nice diagram showing that memory response, we look at a lot of antibodies because they're relatively easy to measure in individual T cells, not so much. So I think we'll learn a lot more in the coming months about what a good immune response is to the virus. We'll also learn more about, does vaccination prevent transmission? And then, kind of more the latter really informs what is life going to look like. So even if we have those vaccines as, kind of a booster / second shot, later this year, next year, I think we have a much better handle on the pandemic and the safety practices we need. So I see a gradual kind of reversing of those, those policies we had where, businesses had to be restricted, schools had to close, and people had to wear masks. I think we'll eventually get to the point where those slowly get rolled back as more and more people get vaccinated and the community rates are lower. I think importantly, we have, at this point compared to last year, we have a better approach at testing, right? We can identify people, that are symptomatic or in some cases asymptomatic. So we have a better sense of, think where we were a year ago. If we saw one case, we'd be freaking out. Now, if I have a case on campus or in school, it's still concerning, but there's an approach taken to make sure that that case doesn't spread across the entire school or the community. So I think it's just kind of this gradual, going back to, okay, I don't think life will be, ever back to 100% normal what it was pre-pandemic, but I think we'll get pretty close where we can kind of enjoy the things that we're used to doing. As more people are vaccinated, I don't know about you, but I haven't hugged my mom in a year. So it's like, I'm looking forward to just hugging, relatives, and you feel safe about it, and to the point where you don't feel like, every situation you go into, like the coffee shop, or if you're driving by a gas station, like everything is like a risk management, right? Like, do I need that cup of coffee? Am I risking my life getting it? So it's just being able to kind of mentally and emotionally get to that stage where you feel less threatened on a daily basis. I think we'll get there, and vaccines are, as Chris and Sarah mentioned, we have to get there. And as long as we can convince people that they are, the confidence is there, increase the confidence that they're safe and effective, the quicker we get there.
Sarah, is there anything you'd like to add about what it looks like for the future for vaccination around with COVID?
The only other thing I was thinking was a little bit about herd immunity. I know we talked about it earlier, but there's no magical mathematical algorithm to figure out preemptively what the magic number is to reach herd immunity. It's something that is calculated in hindsight. So once we vaccinate enough people that we start seeing this reduction in infection, that's then where we say, okay, we have herd immunity. So I think it'll be really exciting as we vaccinate to reach that point. And I think that will be a milestone in this pandemic that will just, I think, give everyone a leap of hope for the future. And then also a goal point to maintain, to make sure that we maintain, once we find herd immunity, once we reach it, to then maintain it and not let vaccine rates drop below it, so that we can keep the pandemic in check.
Well, I want to be mindful of our time while we're here, but I think I want to go back to one of the first questions that was submitted through the chat, and it's kind of how we wanted to think about trying to end the session for tonight, which is, what do we do when we're talking with our friends, colleagues, neighbors, fellow community members about vaccination, and what happens when you face someone who has similar questions like a lot of folks had tonight about some of these different areas, and how do we talk with folks that are hesitant and are leery about getting vaccinated, and maybe even are completely anti vaccination through perhaps conspiracy theories that they've heard. How do you approach that, and what would you recommend other folks to do in that situation? And I'll start with Chris.
So that's by far the hardest question you've asked me tonight. And I don't, I don't really have a real answer for that. I think that, the best that we as scientists can do is try to put out in the most neutral way possible the most accurate information that we have. Because I wouldn't say that I should be up here telling people what to do. I think people need to make their own decisions, but based on the most accurate information that's out there. So, from my perspective, I'd be mostly focused on trying to provide accurate information. One thing that I'll put up, I will go ahead because Sarah's putting me to shame on the slides here. Let me just share this really quickly. This is a very, very simple slide here. But I think, it's difficult to talk to people about risk because people think, well, any risk at all is something that's unacceptable. But the reality is that nothing is without risk. All our lives are filled with risk. Every single thing that you do carries some level of risk. But this just illustrates the fact that, so if you get this vaccine, the risk of the most serious reactions that we know about, that we feel confident are related to the vaccine, these anaphylactic reactions, is less than 1 out of 100,000 people. Okay, there aren't any deaths that we know of that are related to the vaccine. But if you don't get the vaccine, you have hundreds of times higher risk of not just getting infected, not just getting hospitalized, but dying. Okay, so this is based on deaths in New York State, since that's probably the most relevant for this audience, last week. That's the last week's data. So, the point is, if you're balancing risk of getting vaccinated or not getting vaccinated, your risk of death is hundreds of fold higher, hundreds of times higher if you don't get the vaccine than any kind of reaction that you might encounter if you do get the vaccine. So that's the way I would put it. And I don't really, to get back, I mean, I'm really not answering your question because I really don't have an answer to your question.
Well,
and
I really appreciate this. I think this, a lot of folks come at it from a lot of different, there's a lot of hesitancy for a lot of different reasons. And I think you're offering a way in which you could maybe help communicate and break through that barrier when it comes to hesitancy about whether, if they're concerned just about the vaccine in general and what that might do to them. This is quite the illustration about, taking and getting it versus, what the chances are if you don't and you get infected. And, it's not even, like you said, it's not even your risk of getting infected and your risk of having symptoms, but your risk of just death. I think that's a really interesting, illustration to bring to folks. But I don't know, Eric, in what ways have you talked with folks and their hesitancy, that is, is it just risk management, or is there other ways in which you try, with talking with folks?
Yeah, as Chris mentioned, it's a difficult topic because, as you mentioned, Patrick, there are so many different variables and people could be hesitant for a vaccine. I think that's what we worry most about. There are truly people that are anti-vaccine together, but it's kind of that middle gray area, those people that are hesitant. And it's really, I think the important things is one, to hear them out, to listen what their concerns are. Is it risk? Is it that they feel like the trials were rushed instead of just being rapid? So I think it's listening to their concerns. In some cases, it's a concern that might be there for a long period of time. Maybe they had, they have a distrust of the healthcare system or the government or of science. So it's finding out where their distrust lies. And I think Chris's point is great in that you have to give them the information and let them decide. Telling the person that they don't know what they're talking about, they're stupid, isn't going to help the discussion. What you really have to do is just provide them sources of good, trusted information and let them kind of digest that. And unfortunately, this kind of topic is pretty charged. It can take time. You have to give the individual time. And unfortunately, during a pandemic, we're trying to get to an end of. Time seems the thing we don't have. But this is something of a topic that it's not only important just for now of COVID-19, but it's important for other vaccine preventable diseases. As Sarah mentioned, keeping that herd immunity, and we have other vaccine diseases to worry about. So it's really a good opportunity to engage someone and try to hopefully engage in a good two-way conversation, hear what their concerns are, point to some information. And they don't necessarily have to read the clinical trial data or a science paper. There's some good sources out there from reputable news sources that have charts like Chris's that just really put it in plain speak. Why should I get vaccinated? And just try to find that one common area and try to reach that person. But know that it might take some time, unfortunately.
Sarah, I know you do a lot of this locally with folks, and you talk a lot of times with students when it comes, to this in your role at North Country Community College. But what do you do when you talk with folks about this question?
Like Eric, I think the key is patience and empathy, because the concerns come from a real place of anxiety for people, whether they are hesitant. Generally, when they're hesitant, the anti-vax community, I almost think, has a different animal. These are propagandists that are spreading misinformation, which is not the same as someone who's not sure if they want to get the vaccine, or even the average person who is, I'm not getting the vaccine. That's not, to me, an anti-vaxxer. That's still someone who's hesitant. And so that's who I think we're really talking about. So I wanted to share a little anecdote that I was thinking while other people were talking. So my daughter,
Right, Chris.
8, and we went outside the other day, and she didn't want to walk in the snow because she was afraid of snakes. She has a fear of snakes. And, I tried to explain to her, well, there's no snakes in the winter, they're hibernating, there's no snakes in the snow. Even if there was a snake here, it's not going to be poisonous, it's not going to bite you. Like, trying to give her the facts. And she's like, I know, Mom, but I'm still scared. And I think, to me, that's, a lot of times our fears are really, they're founded in our brains. And even if we have the facts, sometimes that doesn't make us feel better. And what makes her feel better is more of a hug and it's okay, you can be afraid of snakes, I'm here for you. And so I think it takes time, like Eric said. Don't judge people, don't write them off as stupid if they are not ready, if they haven't managed to mollify their anxieties about something valid, and just continue to be patient and continue to provide information to people who are open to learning, and baby steps.
Well, I really want to thank you on behalf of everyone who's viewing right now, and reiterate the Chamber's thanks for you three being with us tonight, sharing your expertise and your perspective on this important conversation. I also want to thank our co-hosts, which is North Country Community College, the Ticonderoga Area Chamber, and the Malone Chamber. We had a lot of questions about will this information that you shared be available? Some of the graphics you had will be available, and I will provide quickly in the chat my direct email, because I will receive those slides and the information from folks and share that. It's director@slareachamber.org. Folks can feel free to email me, and I will get the information from our panelists and help share that. And we will also have, the recording. We can work on being able to share that with folks so that they can hear information that you folks shared tonight. So again, thank you so much. This has been really wonderful. I think we could have kept talking for another hour on this subject, and perhaps we might have to do another, conversation in the future. But for now, thanks so much, and I hope everyone who's been listening has a great night. Bye now.
Thanks. Bye.
Thanks. Bye.
Thanks, take care.